get started. Want to be respectful of everyone's time today. So one thing I just want to mention is that please go ahead and mute your audio. It is distracting during the talk to have the audio on. You can put any questions in the chat, and Erin had indicated she will address the questions as they come up. So use that. But I want to say it is my pleasure to introduce Erin Shearer. So she is an assistant professor in the Division of Infectious Diseases in the Department of Medicine at Emory University. And she's a director of the Hope Clinic Vaccine and Treatment Evaluation Unit Research Laboratory. Erin received a Bachelor of Science degree, summa cum laude, in chemistry from the University of Arkansas. So she's in the SEC with us Bulldogs. Her passion for HIV research led her to pursue graduate studies in Dennis Burton's lab at Scripps in La Jolla, where she was selected into a joint PhD DPhil program with the University of Oxford in the UK. And so she was the first to do this. And ultimately she spent 3.5 years at Scripps and 2 years in Sir Andrew Michael's lab at Oxford. After focusing largely on mechanisms of HIV inhibition by broadly neutralizing anti-HIV antibodies for her graduate work, she transitioned to cellular immunology research in HIV and HPV for a postdoc in Phil Greenberg's lab at the University of Washington, and in Denise Galloway's lab in human biology at the Fred Hutch. Following her postdoc, she transitioned to lead an antibody discovery team in biotech for a couple of years at a company called Seagen, then the largest oncology therapeutics biotech in the Seattle area. After realizing her passion for infectious disease vaccine research, she returned to the field as a fellow in the respiratory disease branch at the CDC for 2 more years before joining Emory in March of 2020. Currently at Emory, Erin's independent research focuses on understanding the immune mechanisms that underlie durable and protective B-cell antibody responses in order to more reliably design infectious disease vaccines that provide lifelong immunity. The goal of our VTEU-based immunology research is to evaluate immune responses elicited by natural infection or infectious disease vaccines to inform vaccine development decisions. So it's my pleasure to introduce Erin.
Thank you so much, David, for the invitation, and Ken. So let me go ahead and share my screen. Hopefully there will be no snafus. All right, great. Let me get my laser pointer going. Okay. Today I'm going to talk to you about some research that we're doing in my group, really to try to understand the link between helper T-cell responses and neutralizing and memory B-cell responses to SARS-CoV2. Oh, let me see, I need to advance down here. Okay. And so these are the lab members who generated the data. I apologize, I didn't have a picture for Jen. So a brief outline, I'm going to go over the background, the objectives of our study design, some preliminary data, which unfortunately is all I have to share with you now. We've actually just started working on the samples recently, so hopefully we'll have a more complete story in the near future, and some conclusions and future directions. So some background on the disease and the virus. So as most of you probably know, coronavirus disease is predominantly mild to moderate. So here on the pie chart, you can see that about 80% are mild to moderate. About 40% of that to 45% is asymptomatic, with 20% of that remaining asymptomatic for the duration of their infectious period and the course of the disease, whereas about 25% are pre-symptomatic, meaning they start off being asymptomatic, but they eventually get mild or moderate disease. Approximately 42% of cases are severe and 5% are critical. However, that's not equal among all groups. So there are actually differences in COVID-19 case fatality rate by age. As you can see here, the number of cases per age group, or sorry, the percentage of cases per age group in this graph on the x-axis. You can see according to age group, and then, sorry, on the y-axis. And then here in this slide, you can see the case fatality rate. So the percentage of deaths on the x-axis according to age group here on the y-axis. So with increasing age, there's increasing case fatality rate. But that's not the only population that's affected. Older people also, Black and Latinx populations are more at risk for death and hospitalization, and also those who have pre-existing conditions. So those with diabetes, lung disease, or heart disease are also at increased risk for severe disease and death from COVID-19. So COVID-19 has about 3 different phases that I'll break up here. So the first is the incubation period, which is about 5 to 6 days before the onset of symptoms, which initially start off with relatively mild symptoms, so fever, cough, fatigue, and body aches. And then after about a week, that can progress in those who go on to develop more severe critical disease into having difficulty breathing or labored breathing, and also having more severe disease. So this is really in the second week that the more severe disease onset occurs. And as you can see here, so looking at infectivity, so this is a primary case, and so that 5 to 6 days before symptom onset, they actually begin shedding virus. And so that's how actually secondary cases can become infected before they knew that the primary case had symptoms. And so the serial interval is this range between symptom onset in the primary case and symptom onset in the secondary case. And again, just to really highlight that before someone shows symptoms, they have been exposing other people to the virus. And so, as I mentioned before, there are these phases of COVID-19, so this incubation period and the initial mild disease, and then followed by more severe disease in those which go on to develop it. And that's really shown here by what's happening in the body. So again, these initial mild symptoms, which is followed by a pulmonary phase in more severe disease, and then a pro-inflammatory phase. So this is this SIRS, or systemic inflammatory response syndrome. So there's what they call a cytokine storm. So we detect a lot of pro-inflammatory cytokines in the blood, and there's also a pro-thrombotic phase. And so this really occurs in those subjects which have more severe disease. So SARS coronavirus 2 is the etiological agent of COVID-19, and that was discovered in January of this year. And it is most similar of all the known human coronaviruses. It's a beta coronavirus, and in the genus B, which is most similar to SARS, which was, of course, the pandemic occurred in 2003. Its genome is approximately 80% similar to SARS, or SARS-CoV2, as it's now known. Other beta coronaviruses that infect humans include MERS, and then 2 common cold viruses, OC43 and HKU1. There's also 2 common cold coronaviruses in the alpha coronavirus group, which is 22.9E and NL63. And just here's an overview of the virus structure. So on the surface of the virus, there's spike proteins, also known as S protein, membrane proteins, envelope proteins, and then inside, the single-stranded positive RNA genome is encapsulated by nucleocapsid protein, or N protein. So the virus enters cells, and here this is an example of what's thought to be the target cell, lungs, alveolar epithelial type 2 cells. So spike interacts with ACE2, which is the receptor on these cells. And after this initial binding and attachment interaction, the spike protein is further proteolytically processed by either TMPRSS2, which is a protease on the surface of cells, or by cathepsin proteases that are inside endocytic vesicles if the virus is internalized. And so it's through or after, as a result of this proteolytic processing, that the virus can undergo fusion and release its genomic material into the cell. So here's a sort of find a resolution of that picture. So here's ACE2 on the surface of the host cell, and it actually interacts with a specific domain of the spike protein, the receptor binding domain.
Depende de cada hospital, porque por ejemplo tenemos un amigo en San Antonio que es cardiólogo y a él ya le toca este viernes. Y en el método...
Everybody mute their audio because we're getting some background.
De COVID. O sea, no a los médicos generales. ¿Viste? Cada hospital pone su propia prerrogativa. En el caso nuestro son médicos que tratan pacientes con tumores hematológicos. Primero. Y el personal de mantenimiento.
David, you may be able to do it by yourself, since you are the...
Tienen, digamos, tareas más de dirigencia, que no ven pacientes o que no tienen pacientes con cánceres hematológicos. En ese caso,
can you mute up,
65,
the other attendees?
Así que todo dependerá de quién se quiere vacunar.
Yeah, we are working on that.
Primero.
There we go. There we go.
Okay.
All right, thanks.
Thanks. Okay. So, so ACE2 on the surface of the target cell interacts with the receptor binding domain, which is a domain within the spike. As you can see here, this is the structure of the spike trimer. So each of these colors, this light blue, purple, and yellow, are different units of S protein in the trimer. And this, this is the S1 domain here at the top, and this is the S2 domain at the bottom. As you can see, this shows where the RBD is folded down, and this shows where one RBD unit of the spike is up. So this is where it would be interacting with ACE2 on the target cell. And as you can see here, sorry, I need to move my little Zoom thing around a little bit. As you can see here, the SARS-CoV2 has most homology as far as its amino acid identity to SARS-CoV2 receptor binding domain, but otherwise it doesn't have a lot of homology with the other human coronaviruses, and that'll be important. I'll come back to that later. So the changes that really occurred between SARS-CoV2 and SARS-CoV within the spike that are really notable is that of the 6 residues that are thought to be really critical in the interaction between the receptor binding domain at ACE2, 5 of those 6 residues were mutated in SARS-CoV2 compared to SARS-CoV1. And also SARS-CoV2 has this novel furin cleavage site that was inserted between S1 and S2. So after we have attachment, after we have fusion, after the genome is released, ultimately what happens is that the genome is replicated and the structural proteins are expressed in the endoplasmic reticulum. And this is where within the ER-to-Golgi intermediate network that the budding and assembly of the virus occurs before it's released. And again, I'll come back to this part of the virus life cycle later. So some key questions at this point, I think for everyone, is to really understand how long people will be protected until they become reinfected. And there's not been a lot of reinfections that have been coming up. I mean, there have been a few here and there, but it's not like everyone who was infected back in December or January are all becoming reinfected again with this second wave. Of course, in some respects, those people that were infected in China, which I think has maintained relatively still good control over their outbreaks. So really the question for me as an immunologist and for others in immunology is, what is the duration of natural immunity? And of course, our mild or severe cases, is there a difference in their susceptibility to reinfection? In other words, is there a difference in the magnitude of the duration of immunity between mild versus severe cases? And now that we have vaccines, really the question is, how long are people going to be protected from vaccines until they get reinfected again? So what's the duration of vaccine immunity? Will we need a booster? When will we need a booster? And how does this differ compared to natural immunity? And then of course, you know, or does this differ? And then of course, why? So how does it differ? And of course, I think if we understand ultimately that the vaccine doesn't elicit as good of immunity compared to natural infection, which is how it usually goes, then we would of course want to understand why not, and then hopefully potentially improve the vaccine to make it more similar to natural immunity. At least that's the ongoing goal in vaccinology. Okay, and so just a little background now on the immunology. So neutralizing antibodies block SARS-CoV-2 entry. And we know that neutralizing antibodies are predominantly directed against the receptor binding domain. So this is a figure from Dennis Burton's paper earlier this year, which showed that although most of the binders are directed against the spike protein and a much smaller proportion are directed against RBD only, the proportion of those binding antibodies which neutralize is actually much greater in those antibodies cloned from B cells that bind to RBD. So really it's thought that most of these antibodies which are able to neutralize the virus really block that interaction between the receptor binding domain and ACE2. So we know now through a number of different studies that neutralizing antibodies correlate with protection. And one of the earlier studies that saw this epidemiologically is a really great story, actually. So it was published in the New York Times, evidence that antibodies block the coronavirus. So actually on board this boat, there were 122 people. And what's really phenomenal is that they had serum from these people before they went on this trip. And someone was infected and spread the virus around this boat while they were on their trip. And so of those that got infected, 103 did not have neutralizing antibodies. But of those that were not infected, 3 of them did have neutralizing antibodies in their serum. So again, before they got on the boat. And this was highly statistically correlated with protection. So having neutralizing antibodies was very statistically correlated. It was with protection. But then if we want to take a more sort of isolated experimental model, we can actually show that neutralizing antibodies are sufficient for protection in animal models. So in this case, they passively transferred 2 neutralizing antibodies into non-human primates or an isotype control. 3 days later, they challenged with SARS-CoV2, and they showed that in those which received the neutralizing antibodies, all of the animals were protected from becoming infected, whereas those that received the isotype control showed viral loads both in their nasal swabs as well as in bronchial lavage fluid. So then the question becomes, how long do these neutralizing antibodies last? So this is a paper by Shane Crotty's group where they're looking at antibody levels. And so these are against spike, this is spike IgG levels, receptor binding domain IgG levels, and neutralizing antibody levels. And this is actually, to my knowledge, the study that's looked the furthest out to date and in a very large cohort. And so they show that at 6 to 8 months post-infection that 90% of people are still seropositive for spike antibodies, receptor binding domain antibodies, and importantly, neutralizing antibodies. But what's critical to note about this study and those moving forward is really what the makeup of that cohort was. So you can see here that almost all of the cohort was mild to moderate. So the question is, of course, how does this look like in those which have severe disease? And so in the future, you'll see this little symbol at the bottom. And so that's how I've noted what the cohort makeup is for these different studies. So here's a study which looked at a more evenly distributed severe versus mild cohort population. And here you can see that actually there are much higher levels of neutralizing antibodies in those which have more severe disease. And although it seems from the study that there's no difference in the kinetics, because you have a much higher level in those which have severe disease, the antibodies, as they are slowly waning over time post-infection, it appears that they're staying above baseline here, or above the level of detection for much longer than what they're observing in people who have had mild infection. And a similar result was found in a very recent paper by Scott Boyd's group, where they saw that in 118 inpatient samples versus, so with those with severe disease, there's outpatient samples that they see much higher levels of neutralizing antibodies than those which have severe disease. So this is leading to the question of, do those who have mild or moderate disease, are they going to become more susceptible to infection sooner? And so some early data from one of the vaccine candidates, this is the Moderna mRNA vaccine. This is really only a month post boost. So not really longitudinal data at all in the antibody response, but what we can learn from this data is that the spike and receptor binding domain antibodies are actually much higher than what we see in natural infection by 1 or 2 logs. But what's interesting is that the neutralizing antibody titers are very similar to what we see actually in mild to moderate infection. So not a lot different as far as that's concerned. So in the case of mild or moderate disease, if you lose these neutralizing antibodies, then, will you be immediately susceptible to infection? And we don't know that, but actually we think that there are backups in place through immunological memory. And there's more than one arm of the immunological memory. One of those is cytotoxic T cells, which I'm not going to describe today, which are very important. And then also memory B cells. So memory B cells are B cells that were previously activated. They still express their B cell receptor, which is like a membrane tethered antibody on the surface of their cell. And they're quiescent, and they float around in blood and secondary lymphoid tissues, really waiting to encounter their antigen again, in which case they become activated, they rapidly expand, and they differentiate into these antibody-secreting plasmablasts. And this is really the basis for booster vaccines, because those memory B cells will become plasmablasts and then rapidly boost the antibody response. And this occurs very quickly, so 4 to 7 days post-infection. And what's really important about that, thinking back to those earlier slides, is there's like 5 to 7 days that it's thought that people are really infected before they start to show symptoms. So it's possible that these memory B cells could elicit antibody responses that might even protect before the virus titers really get very high and before it starts to cause disease. So then, of course, the question is, what's the magnitude and duration of memory B cells in people who have had COVID-19? And so again, looking in this mostly mild to moderate population, we see out to 6 or 8 months that there's actually very good levels of both IgG spike-specific memory B cells and receptor binding domain-specific memory B cells. So that's very promising. Again, we don't know in the case, in severe cases out to this, to such a time point. So it really leads us to wonder whether or not the memory is the same in that population. And what I'm very interested in knowing as others is, can these differences in the magnitude of the antibodies or the durability of the neutralizing antibody response, and also potential differences in the memory B cells between severe and mild or moderate COVID patients, can they be explained by T follicular helper cells? So what are T follicular helper cells? So very briefly, in a T cell dependent immune response, B cells, which are responding to the antigen, interact with T helper cells. So these T helper cells express a T cell receptor, which binds a peptide that's expressed by MHC2 on the surface of MHC class 2 molecule on the surface of the B cell. And it's through these cognate interactions that a germinal center is formed. It's thought that that actually through some of these initial interactions that these early activated B cells can become rapidly differentiate into plasmablasts as part of what they call extrafollicular response. And so this, again, I will bring up later. But some of these B cells and T cells are, through the interactions, again, form this germinal center, which is found in your lymph nodes and secondary lymphoid tissues, as well as your spleen. And through their interactions with follicular dendritic cells, which carry the antigen, in which case the virus or the vaccine, these B cells undergo affinity maturation of their B cell receptor and also their antibodies. And they go on to differentiate into long-lived plasma cells, which hone to your bone marrow and express antibodies in your serum that make up your steady-state antibody levels. And they also go on to differentiate into memory B cells, which, as I previously discussed, are really circulating throughout your lymphatics, waiting for their second moment to shine. So they have looked at circulating T follicular helper cells, which are what's the word I'm looking for? They're highly related to germinal center T follicular helper cells. So the germinal center T follicular helper cells are only present in the secondary lymphoid tissues, but the correlate of those in blood are circulating T follicular helper cells. And they've looked at these and found that in severe cases, actually these circulating T follicular helper cells express more inhibitory and cytotoxic genes than those in mild cases. So you can see here the difference in the number of circulating T follicular helper cells was not different between those which had mild versus severe disease, but those which had severe disease had more of the gene expression associated with cluster 5, which includes genes expressed such as TIGIT and LAG3, which are known to be inhibitory. So associated with, for example, exhausted cells, so like exhausted B cells in this case. exhausted T follicular helper cells in this case, so potentially not providing help to the B cells. And also, although not statistically significant, they also found that in severe subjects that they had these cytotoxic TFH. So the question really became, in severe subjects, are these TFH either not helping their B cells, which is really critical for this effective germinal center function, or are they killing their B cell partners? And so actually there's some evidence from that. So in this really, really fantastic paper by Kaneko and colleagues, they looked at lymph nodes, as I'm showing here, and also in spleen of cadavers who succumbed to severe COVID-19 disease. And what you can see here is this is in a non-COVID-19 lymph node. And you can see these really beautiful germinal centers, this really nice organized structure. This is how it's supposed to look. And then if you look over here on the left at the late lymph node, so this is late in COVID-19 disease, and it's actually the same for early in COVID-19 disease. The lymph node, you can see, you don't see any of that really well-defined structure. So really the germinal centers are not there. And the structure is really been destroyed that is normally present. So going back to this, we see that there's higher levels of antibodies in severe subjects. So is it possible that they're coming from these plasmablasts? Because at this point, we know that this is not really occurring in severe disease, unless, of course, we find, and this is an open question, that these germinal center events are actually happening, later. But right now, it appears that this is not happening. So the question is, will these antibody levels more rapidly decline in severe patients over time because they don't have these long-lived cells in their bone marrow, or will they have fewer memory B cells in the long run, because due to the fact that these germinal center structures were disrupted? So these are sort of the open questions that we're interested in learning. So the objectives of this study were really to assess the magnitude and the quality of TFH responses in mild and moderate versus severe cases and following vaccination, and to test whether the magnitude or the quality of the T follicular helper responses is associated with the durability of neutralizing antibody memory B cell responses. And really what we're hoping to impact from this study is to potentially inform vaccine development and or implementation. So learning more about the durability of these responses, will people need a booster if the vaccine responses aren't as robust, et cetera. So some questions, specific questions that we hope to answer. So what is the half-life of SARS-CoV-2 neutralizing antibodies following mild, moderate COVID versus severe COVID or vaccination? What's the half-life of the memory B cells, again, in these separate populations? Because we know those may be an important factor in long-term protection. And are there differences in the magnitude or the quality of the germinal center T follicular helper cells or circulating T follicular helper cells elicited by mild, moderate, or severe COVID versus vaccination? And can we again, see that there's a difference that if you have, for example, a difference in the magnitude of the T follicular helper cells, is that associated, for example, with less disease severity? So if we see more T follicular helper cells, do we see, is that occurring in those people who didn't really have that destroyed germinal center structure? But again, we may see that there's no difference in magnitude, but we may find is that there's difference qualitatively. So do those in more severe disease, again, do they exhibit these markers of either exhaustion or markers that they're cytotoxic? And so we're trying to take a look at that, not just these circulating T follicular cells, but also in the germinal centers. And again, comparing that in mild versus severe disease and then to vaccination. And then ultimately, can we relate those T follicular helper cell magnitude and quality to the durability of neutralizing antibody and memory B cell responses? So very briefly, the study design, we have a mild moderate cohort of approximately 30 persons, a severe COVID-19 cohort of approximately 30 persons. We're still enrolling actually in this cohort. And in these subjects, we're hoping to take two blood samples in their acute phase after symptom onset, and then also obtain a lymph node fine needle aspirate. So that's a relatively non-invasive procedure, which is like a needle biopsy. And so that allows us to sample the germinal center in their lymph node. where they identify the lymph node through ultrasound. And then looking again in the blood at immune responses a month, 2 months after symptom onset, 6 months, and then a year. And then also in 30 subjects, which we're defining as SARS-CoV2 naive, which are those that don't have antibodies to either, principally to the receptor binding domain of SARS-CoV2. And so after they receive this vaccine, we'll be taking blood sample before vaccination, a week and 2 weeks after, again, a similar timing for the FNA, and then before the second dose, the booster dose, assuming that this will be the mRNA vaccine, a week after the booster dose, at a month after, and then 6 months and a year. And so I'll come back to this, actually, I think in the next slide, about this receptor binding domain and why we're using it, antibodies to receptor binding domain, and why we're using it to define those who are SARS-CoV-2 naive. And then also we're using some healthy controls, some of which I won't be showing today, especially for the cellular assays. We're saving them. But these are PBMCs that were collected, so pre-COVID. So these are 2019 and before in 10 subjects, and also healthy serum that again are antibody negative. So we have to be mindful about pre-existing immunity to SARS-CoV-2 because there's evidence that it exists. So importantly, I and some colleagues showed that receptor binding domain is actually exquisitely specific to SARS-CoV-2 in serology tests. So if I or someone else has been infected with a common cold human coronavirus, it's unlikely that our antibodies will bind to SARS-CoV-2 receptor binding domain. So that's good. So this is really a nice marker of whether or not someone has been previously infected with SARS-CoV-2. In contrast, Weck et al. Showed that if you clone antibodies for the memory B cells of a SARS survivor, that those antibodies, some of those were cross-reactive with SARS-CoV-2 and could neutralize it. So there is some evidence that at least in SARS survivors, and possibly also in those who have memory B cells to the human endemic coronaviruses, that there's cross-reactivity in this memory compartment. And Grafani et al., so this is Shane Crotty and Alex Sette's groups, show that approximately 50% of unexposed individuals, so these were PBMCs taken pre-pandemic, so 2019 and before, that approximately 50% of these people have T cells specific for SARS-CoV-2. So there seems to be a lot of conservation between the T cell epitopes in the common cold human coronaviruses and SARS-CoV-2. So again, why we have to be careful in what controls we're using to really understand what our set point is versus really identifying someone who's been infected with COVID. Sorry, SARS-CoV-2. So the preliminary data that we've been able to sort of establish our assays. So initially, I'm going to go through our antibody tests, and because this is an antibody interest group, I'm going to explain the assays in a little more detail. So this is looking at receptor binding domain, IgM and IgG responses. So this receptor binding domain we received from Jens Remmert and his group here at Emory. So this is an indirect ELISA where we coat RBD on the plate, and then we bind with serum or plasma, and then detect with anti-IgG or anti-IgM. And as you can see here on the y-axis is the absorbance, both for the IgM and IgG, and then on the x-axis is the serum and plasma dilution. And our healthy control plasma here, or serum, so this is a pooled sera from 6 donors. You can see that it's much lower than the binding that we're observing in subjects which had COVID-19.
COVID antibody.
And you can see here the distribution of our cohort based on mild, moderate versus severe. And also, as you can see here, CR3022 is a SARS monoclonal antibody that's cross-reactive with SARS-CoV-2. So it's showing that our positive control is working. And so if we look at the antibody titers by EC50, by curve fitting, you can see that there's higher antibody titers higher IgG antibody titers than IgM antibody titers. And that's really true before and after infection. So you can see this rise in acute infection and starting to, and immediately after in convalescence. And then also the same for IgG, where the levels are overall higher. And really, this is a very small subset, so I don't want to make too much of it, but here there doesn't seem to be a lot of difference between those which have, as far as their levels, between those which have severe and those which have mild, moderate disease. But again, that's been shown well by groups which have a much larger number of samples, which we just haven't analyzed them yet. And so here, again, this is looking at ELISA to look at, in this case, the spike antibodies, both IgM and IgG. So in this case, we're using a monoclonal anti-His antibody, and then we're capturing this spike. So this spike was produced at the biofermentation facility, David's facility at UGA. And we received this construct from Jason McClellan's group, which published the first structure of the trimer in Science. And so this trimer has been stabilized with 2 proline mutations, and the proteolytic cleavage sites have been mutated so that it can maintain this pre-fusion trimer formation. So it's most like the native structure on the virus. And then we add serum and then the anti-IgG, IgM secondary. And again, similar to what we saw with RBD, we see that in this case, all of our subjects have IgM and IgG antibodies to spike. We see that our positive control is working. And so everyone here is seropositive, and our negative controls are very low, which is, as we would hope, as we should expect. And so then again, if we look, we see higher overall titers of IgG antibodies to spike compared to IgM. And again, we can look at the changes over time, and we see that essentially that everyone seems to be seroconverting relatively early, although this person here, the subject may not have at pre-day 10. But we see that essentially everyone is making an immune response to these spike and receptor binding domain proteins. And so one thing that I wanted to bring up, and I'll bring it up actually here and then again later in the talk. So we requested that David and his group at UGA express the trimers with cifeninzine. And this was at the recommendation of Jason McClellan's group. And so cifeninzine actually inhibits mannose. And the result is that you get more of the oligomannose glycans on the surface of the trimer. And the Burton group, Des Burton's group, also provided spike to us, and they do not express theirs with cifeninzine. You can see there's not a difference in the antibody binding. I will come up back to comparing these results in the context of memory B-cell recognition. But I do want to mention that a friend and colleague of mine published a nice paper showing that actually SARS-CoV2 has a higher degree of these oligomannose glycans on the surface than is expected in the human host. So human glycoproteins don't typically express high levels of oligomannose. They're mostly complex carbohydrates. But as you can see, like in the case of HIV, approximately 60% of the glycans on the surface of that glycoprotein are oligomannose. And in the case of SARS-CoV2, it's approximately 20%. So the idea from Jason McClellan's group and actually Barney Graham's group is that when in the process of a viral infection, they're making so much of this glycoprotein in the endoplasmic reticulum, that's what's happening is that they're not being properly processed from the oligomannose state to the complex glycans. And so therefore, a lot of these trimers are coming out on the surface of the virus, having a lot more of these oligomannose structures. And so therefore, if you add cifeninzine and you inhibit mannose, then you're really making the glycans appear more similar to the native trimer on the surface of the virus. And so moving now on to the neutralization assay. So we used a pseudovirus neutralization assay. It actually is a lentiviral or HIV backbone. It expresses a luciferase reporter gene and SARS-CoV2 spike on the surface. So when we infect ACE2 expressing cells, in this case HeLa cells, which constitutively express ACE2, then the SARS-CoV2 pseudovirus enters the cells and expresses a luciferase reporter gene. So then when we lyse these cells, we can read out how much luciferase is being expressed with luminescence. And then in the neutralization assay, we add serum or plasma or monoclonal antibodies, and we show whether or not the serum or the monoclonal antibodies can bind to the spike and inhibit this interaction and the infection of the cells. And so there's been a lot of discussion about whether pseudovirus neutralization assays in the case of SARS-CoV2 and of course many other viruses are really reflective of the live virus neutralization assay. And so this is just a couple of examples, but this is from Dennis Burton's group where they looked using these same target cells that we're using. We actually got the cells from Scripps. And they show that there's a strong correlation between using live virus versus a pseudovirus in determining neutralization titers. And they show actually in their case that the SARS-CoV2 pseudovirus was a little bit less sensitive in its readout than the live virus, although other studies have shown the opposite. In this case, this is from Katie Dorr's group, and they're using a very similar pseudovirus to us, so a lentivirus, HIV-based pseudovirus. And in this case, they see very good correlation between wild-type SARS-CoV2 in vitro neutralization assays in the pseudovirus. We do not, however, have the live virus correlation data for our study, so I just want to mention that. So here, looking at neutralization with this assay, we see that all of the subjects' serum and plasma we tested did neutralize the virus with the exception, well, actually not all, but with the exception of 2. And so here to show you is percent neutralization on the y-axis, 50%, so if they reach 50%, they're considered to be neutralizing. You can see that the healthy control serum and plasma, again, that pooled from 5 subjects who didn't have SARS-CoV2 antibodies. 6 who did have SARS-CoV2 antibodies. You can see that they don't have neutralizing antibodies in this assay. And again, the positive control, this is a neutralizing antibody from Dennis Burton's group that he was very kind to provide us with, and you can see that it's neutralizing very well in this assay. So then if we look at the neutralizing antibody titers, again, with the exception of 2 subjects, one which was actually quite early in their response, we see that most of the subjects elicited neutralizing antibody titers. And then if we look at the correlation between binding and neutralization, as other groups, we can show that there's a very strong correlation between receptor binding domain and S-trimer antibodies, which you would expect, but there's known other epitopes on coronavirus spike other than receptor binding domain, which can allow for neutralization of the virus. There's also a strong correlation between RBD and neutralization, and a strong correlation between the S-trimer antibodies binding and neutralization. So now moving on to memory B cells. So measure the frequency of memory B cells in blood and peripheral mononuclear blood cells using an ELISpot assay. And so here what we're going to do is we're going to take those PBMCs, we're going to stimulate them with this mix of IL-2 and R848, which is TLR7/8 ligand. And what that does is basically polyclonal stimulation. It's like a mitogen or a superantigen. And essentially what it's doing is it's stimulating all of the memory B cells in that mixture. So whether it's specific to SARS-CoV2 or tetanus or whatever this individual has seen. And so we get a total readout of the IgG expressing memory B cells in that aliquot of blood, of white blood cells. And then we can, after 5 days after stimulation, we can put the cells onto plates that have been pre-coated, in this case with S-trimer or with the receptor binding domain, and those cells will lay down and they're secreting antibody as they differentiate into plasmablasts from that culture. And so those antibodies will be captured on the surface of the plate, and using a secondary HRP antibody, we can then detect the presence of these spots, which is showing, how many spots are basically equivalent to how many cells were expressing IgG memory B cells to these specific antigens. And what you can already see very quickly is that there appears to be fewer to RBD. And again, PBS is where we didn't coat the plate, but it's been blocked with 10% FBS that's in the media. And so here you can see the memory B cell frequency to S trimer and receptor binding domain. So there's a higher frequency of memory B cells to the S trimer than receptor binding domain. And this may look very low if you're used to looking at antibody responses. But for example, the HPV vaccine elicits approximately 0.2% HPV specific memory B cells. So they can actually be pretty rare, and tetanus toxoid memory B cells are even rarer. So there are not a lot of them, but they do their job very effectively. And you can see that our results are very similar to those from other groups, which also showed a lower frequency of RBD specific memory B cells compared to spike. And so again, looking at this over time, we see that it seems like most of the memory B cell responses are occurring after day 20, which is similar to the results shown by Shane Crotty's group, which is looking after, in this case, approximately day 20, and they see that the memory B cells are coming up against spike and receptor binding domain. So we do see memory B cells from subjects who were severely infected, which is, again, really interesting to see, given that we believe that a lot of these subjects do not have really good germinal center formation. And just back to this trimer situation. So those trimers here, the McClellan trimer, which was incubated with kifinincine at David's facility, were exposed to kifinincine, the cells were during expression. So these have this oligomannose on the surface of the trimer versus the Burton trimer, which should have less of it. And what's really interesting is that, and this is very consistent, we see higher reactivity to memory B cells with the trimers that have the oligomannose on the surface than those that don't. And so what I'm hoping to do with these is use these as bait in flow cytometry experiments, which I've done before in the case of HPV, and pull out these memory B cells using both the kifinincine treated versus non-treated spikes, and then see do these memory B cells have the same frequency of SARS-CoV2 specific antibodies. So in other words, is this specific? And are we getting more SARS-CoV2 specific memory B cells with this particular bait than with a trimer that does not have the high oligomannose? So that's an open question. Okay, so in the sort of last phase of the preliminary data, just to show you that we've looked at circulating T follicular helper responses in a subset of these subjects. And so just to quickly orient you on the flow cytometry data. So we gated these PBMCs first on the basis of not having doublets and being live cells. And then we look for those which have CD3, which is a T cell marker, and are negative for CD45 array. So this means that these cells have been previously exposed to antigen, in this case, hopefully exposed to SARS-CoV2. And then, of course, circulating T follicular helper cells are defined as being, helper cells, so CD4. They're also CXCR5 positive, which is a homing marker to the B cell follicle in the lymph node. And then they express PD1 and ICOS, which are also activation markers. And you can see that these were stimulated with peptides, SARS-CoV2 peptides. We received these from Alex Setti's group. And this was stimulated with DMSO, which is what am I trying to say? Basically what the peptides are dissolved in, vehicle. And then also here below, so these are the same cells from the same subject, but these were not stimulated. So these are stimulated for 6 hours and these are not stimulated. And so you see a difference in the frequency. So for all of these samples moving forward with our cohort samples, again, these are just preliminary samples we're using to establish our assays. We're going to be doing both of these phenotyping experiments so that we can really understand what the frequency of these circulating T follicular helper cells before we manipulate them, and again, what the frequency of T follicular helper cells are that are responding specifically to SARS-CoV-2. So again, you can see overall that almost all of our subjects have circulating T follicular helper cells. So these are activated cells in the blood. And then we can see that over time that there's some even out to past day 30, which is not really typical for circulating T follicular helper cells. Usually in an acute infection or post-vaccination, you really only see them, you know, around day 7 and then they're gone again. And so what's really interesting, and it's not just our group, but other groups, for example, in this publication, show that they're seeing these circulating T follicular helper cells out to day 20. So this is really unusual and a very interesting observation. And so we took those cells that we stimulated with peptides here, again, that we got from Alex Setty's group, or stimulated with the vehicle. And what we can see is that those that are stimulated with peptides express CD40 ligand, which is a protein which provides B-cell help. And it's really specific to those which were exposed to SARS-CoV2. So we see that not only do these subjects have activated circulating T follicular helper cells, but that they're specific to SARS-CoV2. And we see a correlation between the frequency of the circulating T follicular helper cells and the S-trimer specific memory B cells. But again, we don't know much about how durable these memory B cell responses are. And we need more subjects to be able to look at these differences between disease severity that we're interested in. So briefly, conclusions and future directions. So we initiated a study with COVID patients to look at differences between mild, moderate, and severe COVID-19 and their immune responses. We hope to compare these natural immune responses to those induced by a novel coronavirus vaccine in a cohort of 30 subjects. And from our preliminary sample set for patients with mostly severe COVID, we found that most patients elicit neutralizing antibody and binding antibodies to SARS-CoV-2. We find that the serum binding IgG titers are higher than IgM and also correlate strongly with neutralizing potency. We find there's higher frequencies of IgG positive memory B cells against S-trimer than RBD, which others have shown. And we observe prolonged circulating T follicular helper cell responses, which again, others have shown. We were encouraged to find that those who have severe COVID expressed elicited circulating T follicular helper cells that expressed proteins that are important for B cell help. That's encouraging. And we saw a correlation between these activated circulating T follicular helper cells and memory B cell responses. So again, even though there's a lot of heterogeneity in the immune response, we're seeing that there appears to be some common principles. So in our future directions, we're going to be performing this lymph node FNA to really look at the germinal center T follicular helper cell and B cell responses to SARS-CoV-2, which is very important. We're going to be performing transcriptomics on both the circulating and the germinal center TFH because not a lot of studies have looked at both, actually, to knowledge. I don't think any have yet so far, and that's by RNA-seq. We're going to be assessing whether disease severity is associated with the magnitude or the duration of the neutralizing antibody response or memory B cell response, and also examining the durability of the vaccine immunity compared to this natural immunity. And then finally, looking at whether the magnitude or the quality of this T follicular helper response is associated with the magnitude or durability of the neutralizing antibody response or memory B cell response. And so with that, I'd like to thank you for your attention and of course, thank the participants in this study, the group, which is also actually part of my team, which processes samples at Hope Clinic for these huge phase 3 clinical trials we've been running for COVID, as well as smaller studies like ours. The clinical team, which is responsible for enrolling our participants and looking after them. Yen's group for providing us RBD, Alex Setty's group for providing us peptides in these fabulous mega pools against all of the SARS-CoV-2 open reading frames, to Dennis Burton's group for getting us their very beautiful neutralizing antibodies, and also David Damazi's group for getting us the ACE2 HeLa cell line, Barney Graham's group at NIH for getting us our SARS-CoV-2 pseudovirus vectors and plasmids, so we can set up the neutralization assay. And of course, Jason McCullen's group for getting us the spike plasmid. And I'm embarrassed to say that I forgot to add Michelle and David at UGA for expressing the protein for us. So I will have to add that right now. But anyway, thank you, and I'll take questions.
All right, thank you. It's a great talk. So if you have questions, go ahead and put them in the chat. I went ahead and added one general question that I had during the talk. So go ahead and add it there, and then Aaron will address them as they come up. But great talk.
So I don't know, actually, I don't know if anyone knows that this happens. This is not considered normal. Of course, in mice, you can cause disruptions in germinal centers. So you can use a constitutive knockout and time knocking out a specific gene like BCL6 or CD40 ligand, which can really disrupt germinal center function. But I don't know that they've seen this in disease. And that's, so hopefully not to give away our punchline, but I think what's really interesting is that if you look at SARS, people have memory B cells, we know at least out to 3 years. And it looks like they have antibody responses, like 90% of people are still seropositive out to 3 years from SARS. So it doesn't look like their memory B cells and plasma cell are going to be disrupted. So the question is, how are they doing this if they don't have germinal centers there? So maybe it's that these germinal centers during acute infection are really obliterated, but then right afterwards, maybe the germinal center fixes itself and there's still going to be plenty of virus antigen around. I mean, we know that people could be test positive for like 3 months after they've been infected. So the thing is, and I mean that by like RT-PCR, so there's probably tons of antigen around and they're probably thus able to maybe reconstruct a germinal center response later. But that's one thing we're hoping to answer, so.
Thank you. Are there other questions? I'll just ask one more. So are you planning to clone some of the antibody genes? Are you trying to isolate individual memory B cells, or what's part of your plan for that?
So no, I mean, I think it would be interesting to answer the convincing question, right? But I don't, because I think that might be of interest to people, is whether or not we can pull out a higher frequency of SARS-CoV-2 specific responses depending on how the antigen is expressed. But as for our group, I mean, we're a small academic group and we're not going to be competing with the heavy hitters who have already, produced hundreds, if not thousands of really great neutralizing antibodies. And, Laura's group is producing, antibodies that can cross-react to SARS and who knows what other coronaviruses. So no, we're not really in that competition. I've done that before, and I think it's important to do when you're asking a specific question, but really here, we're just trying to look at how long are the memory responses. So at some point, if the ELIspot fails us because you're looking at such a few number of cells, we might then look at enriching cells from the blood and doing flow cytometry to get a better accuracy read of the frequency, but yeah.
Okay. Yeah, one other question.
Yes, there definitely is a mouse model in which the germinal center phenomenon can be studied. So they have mouse models of SARS-CoV-2. And so that would be very exciting to look at. I do not have access to an animal BSL-3 facility. I know that we have non-human primate SARS models. So I think you could look at, yes, the GC problem was in people who have passed away from severe COVID. Yes, that's correct. So like I said, we know that you can disrupt germinal centers ongoing to, like, for example, some model antigen in the midst of a response with mouse models. But to my knowledge, we've never really seen this in humans, but there's not been a ton of studies looking in humans in responses to, like, infections. But in response to vaccines, humans don't have this complete destruction. I mean, it's the germinal center is intact, so.
Oh, just UGA does have an animal BSL-3 lab. We're actually going to be setting up a lab there next year. So if you're interested, you can get in touch with me, Aaron, about that.
Yeah, that'd be great.
Go in that direction.
In the B cell ELIspot, since you stimulate for 5 days, have you checked to see if there's any proliferation of the B cells that can affect your percentages? Yes. So what we do is we take this relative to the total IgG expressing memory B cells. And that's the nice thing about the assay, is that we have this kind of internal control. So we know how many IgG expressing B cells there are. And so we always express that as a percentage. So our specific as a percentage of that. Does that make sense, Scott? Okay. Yeah. So there's, I think it's also important to not just like stimulate cells polyspecifically, but also to actually stimulate them with antigens, so RBD and SARS-CoV2. But then the problem with that, as you point out, is that then we don't have a really nice internal control. So we plan to do some of those experiments, but then we have to be a lot more careful about what frequency of RBD and SARS-CoV2 specific memory B cells are out of the total.
All right, any other questions? All right, well, I'd like to thank Erin for a really great talk, and also ABRF for sponsoring this webinar. I hope you all have a nice holiday, and look for more webinars in 2021.
Great, thank you, David.
Thank you, David. Thank you, Erin. Thanks everyone for joining us. Have a great day.
All right, bye now.
Bye.
Híjole.